Well, students, this is chapter 20, part 1. Part 1 encompasses 3 of the 4 sections in chapter 20. The overall purpose of this chapter is to give students a feel for how tissues are arranged and how they function, and also how things can go wrong within those tissues to develop cancer. Since this is the final chapter in this textbook, the authors bring back a lot of concepts from previous chapters as revision tools and reminders. So one of the things they started off the entire book with was a definition of what life is. If you utilize the Mr. Gray plus, the plus stands for cells and DNA. So cells themselves are pretty squishy and weak, and they can be easily manipulated through mechanical and chemical means to either dehydrate or to overhydrate and burst. So cells can break. But the question is, how do we then explain the great height of redwood trees where the cells at the base of the tree are supporting the entire height of that giant structure? Or in the case of animals, the same thing. The feet of an elephant are composed of cells, and how do those structures, the tissues in the legs, support the weight of an elephant? The answers to those questions are in this first section of this chapter. Most of the conversation will take place around multicellular organisms. And multicellular organisms contain cells into very organized compartments. But this story is not just about the cells, it's also about the material around the cells, their surroundings. So in combination, maybe that's the answer to the question that we raised a few minutes ago. The first figure in the textbook shows us human kidney cells arranged in a circular fashion along collecting ducts. The important thing to realize here is that there are stains outside the collecting ducts in this region here, and this is the extracellular matrix. And the question is, where did it come from? And it appears that these cells and the surrounding cells, they are responsible for secreting this matrix. That has a general name called the extracellular matrix. So tissues can be defined as a cooperative assembly of cells which have a common purpose to give the tissue its functionality. The bodies of vertebrates are composed of 4 types of tissue, believe it or not. There's nervous tissue, there's muscular tissue, and there's epithelial tissue. And all other kinds of tissue are classified into this leftover bucket called connective tissue. So that would include bone, cartilage, blood, and the regions that are between different layers of the other 3 types of tissue. One specialized role of connective tissue is, as the word suggests, to connect the other layers of tissue. And you can see that here in this illustration. This is a cross-section diagram of a human gut and showing just part of the lower section. And you can see the green areas. There's 2 of them, one down here and there's one up here. These areas are connective tissue, and they connect the epithelial cells to the muscular cells in both cases. The main purpose of the section that we're about to tackle is to illustrate how the cells themselves secrete the material around them, which then becomes the extracellular matrix. And examples of extracellular matrix would be the bone that encompasses certain types of bone cells, and of course the wood that makes up the parts of plants and trees that we're familiar with. The section also addresses cell junctions, how cells make contact with each other and with their surroundings. And finally, we'll be looking at the forces which have to be transmitted across cells to make it possible for huge structures like the redwood tree and the elephant to exist. And that should become obvious very quickly. The organization of tissues into organs, into organisms, is pretty complex, and that requires cooperation. And that cooperation is necessary to maintain the cell numbers in a balance so that each type of cell has its own role and it doesn't overwhelm or underwhelm the functionality of its supporting cells. Thus, systems are necessary for maintenance and renewal of cells when necessary. Thus, cells must communicate with each other so that they have some type of organization and coordination as necessary to function properly. This chapter specifically addresses multicellular organisms, of which the vast majority are plants and animals. Plants being sedentary, that means they don't move, they have rigid structures, and the cells, when isolated from those cell walls, are pretty weak with respect to their ability to handle any kind of physical, mechanical, or chemical assault. Animal cells, by contrast, have muscles which allow those animals to move. This form of movement entails a change in the animal's shape, and that change in shape promotes the transmission of mechanical stress forces through cells. The independent solutions that plants have come up with rely on the production of cell walls and extracellular material deposited either in the cell walls or beyond them. Cell walls are very tough, but they're not really rigid. They need to have some flexibility. Each cell secretes its own cell wall, so every cell wall in the plant is produced by one of its cells. Depending on the location of that cell, its cell wall may be thick and hard, as in the example of wood, or it could be thin and flexible, as in the example of cells in the leaf. If the cell wall is stripped away from any plant cell, then those cells become really fragile and they easily burst under mechanical or osmotic stresses. Interestingly, plant cells contain no intermediate filaments. They do contain microtubules, and they do contain actin filaments, but they don't contain any intermediate filaments. So plant cells cannot be stretched. These cell walls that plants generate can be broken up into two categories. The primary cell wall, made initially during development, which is normally thin and expandable, and a secondary cell wall, which is deposited once the cell has reached its full growing potential. And they can become rigid and thicken to become waxy or woody. What's the chemical composition of these very important cell walls? Well, primary cell walls normally have pectin deposited in them as the cell wall is being laid down. And then subsequently, in secondary cell walls, cellulose is deposited at that point. Both of these are polysaccharides, sugar molecules, just like glycogen. But they have a slightly different arrangement of atoms, and that gives them these unique properties. Indeed, cellulose is one of the most important and most prevalent polysaccharides in the world. Water plays a very important role in regulating the behavior and interaction of plant cells. The force exerted on the cell wall by the plasma membrane of plant cells is known as Turgor pressure. And that Turgor pressure is a result of osmotic pressure changing within the cytoplasm of the plant cell. The cellulose component of the cell wall, deposited as microfibrils, provides tensile strength. And these fibers are arranged along stress points that the plant cells experience. Other components, such as pectin and proteins, are deposited as necessary to support and enable these cell walls to resist compression forces, which would otherwise squash any cells inside. The yellow structure indicated in this figure is the middle lamella, and that has a very high concentration of pectin, and that is used to connect to neighboring cells. The cellulose microfibrils are so good at resisting forces that they can prevent a cell from growing in a particular direction. As indicated here, if the blue lines indicate the rings of cellulose that are deposited in the cell walls around the cells within, then there's only one direction in which the cell can grow, which is perpendicular to these rings. So if the rings were relocated in a different orientation, then those cells would only grow in that direction. An unusual finding with plant cells was the way that the cellulose is manufactured. It's not manufactured inside the cell and then exported outside to be polymerized. It's actually synthesized outside the cell with enzymes which are embedded, as you can see here in blue, inside the plasma membrane of the plant cell. So these enzyme complexes, called cellulose synthase complexes, are responsible for taking glucose monomers and then polymerizing them to form cellulose. The other thing that this figure is showing, not too well though, is that the enzymes can travel along the cytoskeleton, the microtubules, within the cytosol of the plant cell. So these tracks are laid down prior, as you can see here in panel B, and the cellulose that's produced, as you can see in panel A, is basically a mirror image of the tracks of microtubules. So the microtubules come first, and then the cellulose is orientated accordingly. Turning our attention now to animal cells. As we know, animal cells do not contain any cell walls whatsoever. So the outermost layer is normally a layer of sugar molecules attached to the proteins of the membrane itself. We are going to concentrate now just on one type of tissue, connective tissue. So recall that there are 4 types of tissue in vertebrates. Connective tissue is a bucket into which none of the other types of tissues fall. So that's going to be nervous tissue, muscular tissue, and epithelial tissue. With respect to connective tissue, those cells secrete a varied type of extracellular matrix. And if you look at the examples that include connective tissue, this should become clear. So bone has a very solid and durable extracellular matrix made up of collagen and calcium crystals. Tendons have a slightly more flexible version. Cartilage is even more flexible. And of course, blood is the other side of the spectrum, being very, very fluid. The most abundant protein that you find secreted into the extracellular environment is collagen. And collagen is in fact one of the most prevalent proteins found in animals. There are many classes of collagen found inside the body, and they appear in different places. Since we just spoke about bone, here's an actual picture of bone showing the cells in black, and the connective tissue is the gray material in rings that you can see between the cells. And embedded in there is calcium connecting and cross-linking those collagen fibers. Let's spend 3 slides focusing in on collagen. So as we just said, collagen occurs in many flavors in different parts of the body, and there are a number of genes making collagen. Regardless, collagen is a protein. So it's made as a polypeptide of a specified length, and once it's made by the ribosomes, these polypeptides then naturally bind with 2 other polypeptides to form a molecule of collagen. And this happens naturally within the cell. The molecules of collagen then combine with other partners to form a collagen fibril. You can see the diameter of the molecule is 1.5 nanometers, and each one of these little green lines that you see here poking out from the fibril and then increases the diameter to between 100 to 300 nanometers according to the type of collagen that's produced. And believe it or not, these fibrils will come together to form collagen fibers. So each one of these black dots that you see here inside the fiber is a fibril. And they can be up to 3 micrometers in diameter. The collagen is laid down by fibroblasts in most tissues, except that their name is osteoblasts inside bone, as indicated here. So depending upon the role and the need, different solutions have evolved for laying down the structure of the collagen and its associated proteins and sugars. As mentioned in this figure, the cornea of a rat eye is shown, and you can see the fibroblasts that are part of the cornea and the collagen interlinking those cells and strengthening the structure of the cornea. It also says in the legend that some of the elements are dissolved away to make the structures more obvious. And that will be mostly the water and, of course, the ions that are embedded in this matrix. One thing that you have not been told yet is how this collagen makes its way into the extracellular environment. And that's what this slide is trying to convey. So the gray matter here is the cytosol of a cell, a fibroblast. There's its plasma membrane. And here we have a vesicle. Even though it's not spherical, it's still a vesicle. So these collagen fibers are made in the rough endoplasmic reticulum. And in the lumen of the rough endoplasmic reticulum, they find their partners and they start forming the molecules. What you may not know is that the collagen is made as a procollagen polypeptide. So it has on its ends extra amino acids which prevent it from fusing with another collagen molecule. And that's the way that these vesicles then transport out of the cell through exocytosis these pre-collagen molecules. Once in the environment, enzymes present, proteases, will then digest off these green areas to reveal the sticky ends of the collagen molecule. And then the collagen molecule will start assembling, as we mentioned 2 slides back, into a collagen fibril and then a collagen fiber as aggregates. The name of these enzymes, which are responsible for cleaving the green areas, are procollagen proteinases. Any part of this pathway of collagen deposition into the extracellular matrix may be defective. In this particular image, the skin of this famous person has a defect in one of those enzymes that cleaves off the procollagen fibrils. In his case, the collagen is unable to form the fibers needed to interconnect different molecules. And therefore, his skin is very, very elastic because of that lack of connection between the individual molecules. You may be aware that certain cells in the body are mobile. They can move around, such as white blood cells. The question is, how do these white blood cells then reach the destination they seek? And the answer is simple. They have enzymes that they secrete ahead of themselves that are able to melt or break these collagen interlinkings, therefore allowing passage of that cell unhindered through that matrix. Once the cell has passed, the fibroblasts are able to repair any damage behind them, therefore fixing the pathway. These matrix proteases are the names of the enzymes that are able to cut the collagen interlinks. That's not the only place that we find the remodeling of the extracellular matrix. So during growth and repair, and also during renewal and disease, these same mechanisms can operate. The fibroblasts, which not only secrete the collagen, but also mold it, have very varied lives. In this example here, the skin of a tadpole, we can see that different layers of the skin have collagen fibers orientated in different directions. So this layer here, where the cursor is coming out of the slide, whereas the adjacent layers on either side are impregnated with collagen fibers going from left to right. In this famous experiment where cells were removed from the heart of mice and then positioned on plates in the lab, the collagen, which is present as collagen fibrils in the environment, have been remodeled by these 2 fibroblasts. These cells move up and down the collagen fibers, repositioning them and laying them in a defined pattern. How then are the strong collagen fibers connected to cells? And that's illustrated in this slide here. So the collagen fibers are here in yellow, and for them to then be connected to the cytoskeleton of the cell requires an interplay of all these proteins. And the most important protein here happens to be this one called fibronectin. Fibronectin. So fibronectin works as a dimer, and along that particular polypeptide are two binding sites, one which binds to the collagen and the second one which attaches to other molecules in the membrane of the cell. And these other molecules are normally integrin proteins. And the integrins, as we learned in the past, are transmembrane proteins which have domains on the outside and domains on the inside. The inner domains are connected to the cytoskeleton, in most cases the actin filaments, but in some cases the intermediate filaments, through a combination of adapter proteins. Any stresses that are placed on the cell are transmitted through this combination of proteins to the collagen fibers, and likewise any stresses outside the cell are then transmitted into the cell through the same interplay of proteins. Concentrating a little bit further on these integrins, humans have more than 24 types of integrins that they utilize in different combinations in various cells in various tissues. One thing to notice is that these proteins, the integrins, have a hinged domain along their length, and they are able to exist in an inactive form and an active form. The inactive form protects the active sites which bind to the extracellular matrix, allowing the cell to move by detaching itself from the extracellular matrix. But when the cell is ready to reattach itself, then the integrins are activated and they unwind, and they are then able to expose the active sites, therefore seeking and connecting to the extracellular matrix. One important point that the authors make is that this is a two-way interaction. So we could have binding to the extracellular matrix, cross-talking through the integrins into the signal transduction system of the cell and changing the cell's behavior. Likewise, we could have the cytoskeleton within the cell cross-talking outwards into the extracellular matrix. And that's what these two black arrows are trying to convey to us. That bigger picture is relayed in the top half of this slide, and the overall meaning that transgresses lots of chapters is indicated below. That the cell's external environment is responsible for deciding if the cell lives or dies, and if it does survive, whether it grows, divides, differentiates, or stays the same. So the extracellular matrix is more important than you may realize. As was alluded to a number of slides back, the extracellular matrix is not just made of proteins. The second most common constituent are sugars. But these are not your regular sugars, these are modified sugars. So one of the most common are called GAGs, G-A-G-S. The GAGs stand for glycoaminoglycans. So glyco means sugar, amino means like amino acids, glycans means sugar. So these molecules are mostly sugar, even though they have the odd amino acid attached to their side chains. These covalent linkages to the proteins can result in the formation of absolutely huge molecules whose purpose is to protect the cell and to carry information, such as for the immune system to leave these cells alone. Let's turn our attention to section 2 of this chapter, which explicitly talks about epithelial sheets and cell junctions. Epithelial sheets are very, very common in animals because they form boundaries between tissues and organs. There's two important points to learn about epithelial cells. The first is that they come in two varieties. They can be simple epithelial cells, where they're just one cell thick, or they can be stratified, in which they are multiple layers thick. And examples of each are present all over your body. So this would be an example of skin cells, epithelial skin cells. And this would be an example of digestive cells lining your digestive system. Now within each of these classes, the cells can have different shapes. Some of them could be flat, some of them could be cuboidal, and others could be very tall. and that can exist at any point. The important point for you to realize is that just like a plasma membrane has a covering that protects the cell and it allows many functions, the same thing can be imagined for a multicellular creature where these linings can be seen as plasma membrane equivalent. And they act as barriers, they allow transport between the chambers, and they regulate the flow of chemistry. They allow one side or another to communicate, and they also perform protective functions. A very important observation when it comes to epithelial cells is that the circumference of the cell is not equivalent. These cells have at least two surfaces. They have an apical surface, which faces the air or a liquid, and then they have a basolateral surface. And in this case, it starts at the thick line and it goes down across the bottom and then up again, up to this point. The basolateral surface may have different chemistry in terms of proteins and lipids in its membrane compared to the apical surface. And that's going to be very important as we discuss in a couple of slides. Now, these cells are attached to each other through tight junctions, which we'll talk about again in a moment, but they are attached to a very strong piece of connective tissue called the basal lamina. Not only is the basal lamina important for attaching the cells and giving them shape, it's also important for communicating information into these cells. The epithelial cells composing the digestive system, the human gut, are an example that's used by many textbooks to illustrate many facets of how epithelial cells work and how they're arranged. The goblet cells, or otherwise known as mucous cells, their internal architecture is completely polarized. Their nuclei are distributed towards the basal surface, whereas their Golgi is always orientated facing the apical surface. And so are their secretory vesicles, which are packed facing the membrane of the lumen of the digestive system. This makes sense because once the polysaccharides, the mucus, is made, it's stored in these vesicles and it gets secreted in this direction upon need. Likewise, the absorptive cells, the ones in gray here, their apical surface is full of proteins that can absorb digestive products, and their basolateral surfaces possess alternative chemistry for moving substances into the bloodstream below. The function of the tight junctions is paramount in preventing the mixing of different types of plasma proteins and lipids. We now turn our attention to the cell junctions which exist, concentrating first on the tight junctions as we just looked at. So this schematic is showing you how the cells lining the digestive system and other epithelial cells have evolved. The primary purpose of tight junctions is as a sealing agent between neighboring cells. They are not there to prevent the passage of chemistry between the cytoplasm of cell 2 and cell 3, but more to prevent the passage of extracellular material from one side of the cell through to the other. So if you look very carefully, these yellow tracer molecules are present only in the brown region, but they cannot pass between the cells into this blue region. And that's very important. Otherwise, food molecules like glucose in the blood would simply diffuse out into the gut and would lose all our energy. One can imagine the same situation within the bladder, where urine in the bladder could leak back into the body, into the blood. Images such as this prove the resilience of tight junctions. So here we have a dye that's been applied to the lumen surface of the epithelial, and that dye is unable to pass into this space here through the tight junctions. And the same experiment done in reverse, where the dye is now applied to the blood side of the cells, that dye is prevented from leaving into the lumen by, again, tight junctions. If we take one deeper look at tight junctions by magnifying the area of the membrane between two adjacent cells, so that the cytoplasm of cell 2 is here where the cursor is, and the cytoplasm of cell 1 is now where this cursor is, you can see that tight junctions consist of proteins very closely opposed to each other in the membranes of the two cells. The most common proteins found in tight junctions are occludin and claudin. Just to conclude, that students know this, the purpose of tight junctions is to prevent the migration of proteins serving one function in the apical region of the cell and those proteins serving a different function in the basolateral region of the plasma membrane. In addition, the vesicles which fuse with the membrane do so in the vicinity of the tight junction, that way guaranteeing that this polarity is maintained. So vesicles will fuse up here to deliver the cargo that will keep the proteins in the apical region up here. And then other vesicles will fuse down here, delivering their cargo to add to this membrane below the tight junction. Turning our attention to two other types of cell junctions, adherins and desmosomes. They're very similar, but also different. Adherins and desmosomes share one thing in common. Their purpose is to mechanically bind cells together like molecular staples, so that if you pull on one cell, that force is transmitted across the plasma membrane of that cell into the cytoskeleton, then across the desmosome or adherin of the far side of the cell into its neighboring cell or the extracellular matrix. So they provide mechanical strength in all cases. The main proteins found within the adherins and the desmosomes are cadherins, and they come in many flavors. But there is one difference. Whereas adherins are laid down as a belt around the periphery of the cell, desmosomes are like small patches, dots, like rivets, that are placed in important locations. This figure here shows how both of these types of cell junctions work. So we have the cytoskeleton here, and that could be the microtubule, or it could be actin filaments, or even the intermediate filaments. And they are connected through linker proteins to the cadherin molecules. And the cadherin molecules have a liking for each other, forming what's called a homophilic binding. And in the presence of calcium ions, these bindings are strengthened even further. Here we see the two types of cell junctions, adherins and tight junctions, on the same slide. So here are the tight junctions, and the membranes in these regions are very close together, and their purpose is to prevent fluid from passing between the cells. But here we have the adherins, and you can see the proteins of the adherins cross-linking the membranes of the two cells. If you look very closely, the membranes are the normal distance apart, and the proteins span the gap between them. But on the inside of the cell, there's a ring, in this case, of actin filaments, which anchor to these proteins through adapter molecules. During development, some strange things happen to different tissues as the embryo develops. One of them is the formation of the neural tube, indicated in this figure here. So this tube of cells has somehow formed this U-shaped channel from a sheet of cells. And the second thing that can happen is that these sheets of epithelial cells can pinch off and form spherical vesicles. And that too is indicated in this picture. How does this happen? Well, the actin filaments at the adherins are able to contract, and when they do, they force the cells and the basal lamella to take up the appropriate shape. So changing shape is a role that actin filaments associated with cadherins can play in cell behavior. Let's focus on desmosomes. Desmosomes are like molecular staples, They staple cells together so that forces can be transmitted through their cytoskeletons. In this case, the blue molecules in cell 1 are the keratin filaments, part of the intermediate filament family, and they are connected through these plaques of linker proteins to the cadherin molecules. And on the other side, we have a mirror image of the same structure. Desmosomes offer great tensile strength in that they don't give, so forces are transmitted in all different directions. One of the most common uses of desmosomes is in exposed epithelia like skin cells, where stretching and tugging are very common. Desmosomes can also connect one cell to the basal lamella, and in that case, the other half of the desmosome is missing. So these are called half desmosomes, or more precisely, hemidesmosomes. There's no difference with the half desmosome compared to the full desmosome. If this relationship between the desmosome and the basal lamella is dissolved, then blisters are a common occurrence when these skin cells are either broken or pulled off the surface due to excessive force. There's yet another class of cell junction called a gap junction. These have nothing to do with the transmittance of force. They are more for communication between neighboring cells. The size of the gaps between adjacent plasma membranes is tiny because it's controlled by the diameter of these connexons. Connexons are just proteins of adjacent cells which are lined up so that there is a small passageway between the cytosol of one cell and its neighbor. As it says here, the exchange of molecules is limited to small ions and tiny molecules, no more than 100 Daltons in size. So, although proteins cannot pass through these gaps, ions and small chemicals can. And nature makes use of this, especially in examples cited in physiology textbooks with heart tissue. So when heart cells beat, electrical signals are passed from one cell to the other in a coordinated fashion so that the entire chamber of the heart beats as a single unit. The dynamic nature of gap junctions is beautifully illustrated in figures like this, where the scientists use dopamine to change the behavior of cell-to-cell communication. So panel A shows neurons which are interconnected via open gap junctions, and the same neurons after the administration of dopamine, which restricts these gap junctions from passing material through. Here we have a summary slide illustrating and tabulating the information we've learned so far about the 5 different types of cell junctions. Please spend some time revising this information. Returning quickly to plant cells also have cell-to-cell communication, but in a completely different architecture. So plant cells came up with their own solution and an evolutionary separate lineage from animal cells. In plant cells, these regions that connect neighboring cells together are quite large, and they're called plasmodesmata. Plasmodesmata. Because of the size of these, large proteins can be shared as well as organelles between neighboring cells, as well as regulatory RNAs. Because plant cells are immobile, how do you transmit knowledge from the roots, say to the tips of the leaves? Well, one way is to pass that information through the plasmodesmata of neighboring cells until you reach that location. Focusing on the third section of the chapter, that's to do with tissue maintenance and renewal. All animals in natural situations begin life as a fertilized egg known as a zygote. The different zygotes of different species may look the same at this stage, but they lead to the production of completely different arrangements of tissues and cells. Well, so what happens? Well, if you look back here at the zygote stage, there are 2 major differences. The DNA within each zygote is different, therefore carrying different genes that will be expressed at different times. And the zygote itself carries a repertoire of proteins, mostly transcription factors, that may be different, therefore activating different genes at different times. One of the marvels of development is how do these single cells know where to position the millions and trillions of cells that they will produce, and what are the arrangements in different tissues. So tissues appear to have very complicated yet coordinated structures when it comes to cells. A good example to examine is thick skin. Thick skin has two major regions, the outer epidermis and the inner dermis. The outer epidermis here is basically made of stratified layers of epithelial cells. The dermis, on the other hand, is a collection of loose and dense connective tissue in various amounts. In both, we have blood vessels and nerves as well as muscles. So skin is pretty complex. However, this is not an exception, it's more the norm. So once you study enough tissue, you come to some very simple conclusions pertaining to this section. So in order to preserve the organization of tissues during development and into the adult, it's very important that there's cell communication. Through cell communication, cells can talk to each other and control each other's behavior and fates. The second thing is that cells will only stick to what they're supposed to stick to, and it prevents the tissue from adopting a random arrangement. So if you are a cell that is looking for the basal lamella, then you will only stick to the basal lamella and not to another cell. Number 3 is cell memory. As cells divide, they don't have to go back to their foundations and relearn what they're supposed to be. So as a mother cell divides into daughter cells, the DNA program is passed intact, and that's cell memory. We covered cell memory in a previous course. Another component that students must recall is that cells live for different time periods. So nerve cells will last a lifetime, whereas intestinal cells will only last 3 to 6 days. Once these cells die, how are they replaced? And the answer is stem cells. These stem cells have the ability to self-renew again and again, almost indefinitely, or at least for the lifetime of the organism. And every time they divide, the daughter cells can either be differentiated down this pathway here, or they can be told to renew the stem cells so that the population of stem cells never dries up. This pathway here leads to, first, precursor cells, which can divide for a limited number of cell cycles and generate daughter cells that are able to become terminally differentiated cells. After development ensues into the child and adult, these stem cells have a limited capability. They can't generate any cell in the body, but just a few types. For instance, they can generate blood cells, but not gut cells. There'll be cells in the gut that can make gut cells, but not blood cells. These gut cells are very good places to look to learn further about the behavior of stem cells and their neighboring cells. This magnified picture taken with a light microscope of stained tissue shows how the arrangement of the villi and the crypts in the gut lining is complementary to the way that stem cells work in this location. These figures here, simply cartoons of the previous slide, they magnify the crypt to show us that the stem cells are located at the base of the crypt. And when they divide, they give rise to these precursor cells. And as more stem cells divide, more and more daughter cells are pushed up, pushing these cells to become more differentiated here until they eventually become part of the villus, and then they fall off into the lumen of the digestive tract. When one compares the way that stem cells and their precursor cells function in the gut, and they compare that to skin cells, there's a sheer difference between the two processes. The alternative anatomy of the skin has the stem cells being part of the basal epithelial layer, the one that touches the basal lamella. And their daughter cells are pushed upwards away from the basal lamella, whereas in the gut they're pushed along either to the left or the right. Regardless, the differentiated cells are further away from the stem cells in all cases. Yet another architecture exists when it comes to the fluid connective tissue, blood. The stem cells are not located in the blood, but they are positioned in bone marrow, and those stem cells can give rise to any constituent when it comes to red blood cells and white blood cells, as indicated here, including the platelets. Further investigation of how the crypts work in the gut has revealed the existence of a protein called WNT, WNT, that's part of a cell signaling pathway that operates. Now we'll see how WNT works in the cancer part of this chapter, but for now it's important to realize that the penneth cells in the base of the crypt alongside the stem cells are responsible for releasing this protein as a growth factor into the crypts. These cells have two functions. They protect the crypts from bacterial infection by releasing other proteins that act as antibiotic peptides, as well as this signaling molecule. The WNT pathway keeps the proliferating cells active, but once they move out of the base of the crypt, then they are no longer permitted to divide. This is a very elegant system of controlling cell behavior. The discovery of stem cells has given much hope to personalized medicine as well as research into diseases. The embryo of any animal develops into a blastocyst. A blastocyst is a hollow ball of cells with a developing inner cell mass as indicated by these pink cells. And this is where the embryonic stem cells come from. If you remove these cells, they can be grown indefinitely in culture, as well as, if treated with certain chemicals, they can be differentiated into almost any tissue of a baby. The exception being the placenta itself, in which this egg is growing. These embryonic stem cells are thus referred to as being pluripotent. Pluripotent. That means they can produce a whole bunch of other cells, not just a limited subset. That ability is what makes them perfect for research purposes. As illustrated here with embryonic stem cells which are forced to become an eye cup. The proof that this structure here in green is part of the eye is indicated here when pink color is produced by staining proteins that are only present in the retina of a real eye. Further understanding of how tissue develops has taken human beings into reproductive cloning methods that are quite common now for husbandry of animals. Many of the cows around us are bred in this fashion. In this technique, the nuclei of adult tissue cells are captured, and then those nuclei are injected into eggs whose own nuclei has been removed. So we have cytoplasm from an egg, but the nucleus from an adult cell. Upon proper stimulation, some of these cells, a small minority, are able to develop into the blastocyst, and at that point they can be then transplanted into the uterus of a surrogate mother, and they result in the production of babies corresponding to the nuclei of that parent. An alternative strategy would be then to use these cells for devising personalized medicine, such as designer organs, because that negates the use of anti-rejection drugs for the life of that individual. And these drugs can be pretty expensive.